analysis of resting state fMRI data and structural connectivity analysis of diffusion weighted imaging data to examine the organization of large-scale brain networks supporting executive functions. Her current projects focus on understanding dynamic networks interactions underlying cognitive ability in neurodevelopmental disorders such as autism. Dr. Uddin's work has been published in the Journal of Neuroscience, Cerebral Cortex, JAMA Psychiatry, Biological Psychiatry, PNAS, and Nature Reviews Neuroscience. And she has received numerous awards, including the Young Investigator Award by the Organization of Human Brain Mapping in 2017. She has received the, the University of Miami Provost Research Award, I think, numerous times, as well as many other awards, including the Brains Award. The title of her talk is Brain Dynamics and Flexible Behaviors, and please join me in welcoming her. I'm going to end the slideshow and stop sharing. So Lucina, you can, if you don't mind putting up your slides again, that would be great.
Okay. Can everyone see?
Thank you.
Thank you.
Very excited to have you. Thank you so much.
Great, thank you. Can everyone see this title slide now?
Not yet.
It's not up yet? Oh, let's see. What happened? Share screen. Okay. How about now?
Yes.
Okay,
Yes.
Wonderful. Okay, thank you so much, Fumiko, for inviting me to do this, what's become the new normal for us, giving talks from our own homes. So I'll try to share with you what I would normally share with you when I give a talk standing up somewhere, but I will be sitting down. So basically, I want to talk to you today about brain dynamics and flexible behaviors. And normally I would, oh, how do I advance slides in this? Good question. Oh, did that work? Okay. Oh, it worked. Okay. So normally I would begin with talking about why we're so interested in flexibility in the lab. And one of the things we study is autism spectrum disorder, where we know there's some symptoms of insistence on sameness and sort of rigidity and kind of wanting to do the same thing in the same way. So a lot of parents or caregivers will say that, well, their kids with autism want to wear a particular pair of socks or a particular kind of clothing. And sort of these inflexible behaviors can sort of interfere with day-to-day activities if they're not under control. And so that's sort of our sort of segue into flexibility more generally. But nowadays, I think everyone is engaging in flexible behaviors as we transition from teaching in the classroom to online teaching. We transition from leaving the house to not leaving the house. We're sort of going through this in our own lives now, and it's sort of acute how much flexibility is required to get through this kind of interesting time in our lives. So I think I don't even have to explain to you why flexibility is something of sort of broad and general interest. So what our lab tends to focus on is sort of how brain networks develop from childhood through adolescence and into adulthood. So we use neuroimaging to study brain network development and how that relates to cognitive development. And also what are sort of the consequences of atypical development of within and between network connections as we go from childhood, adolescence, and into adulthood. Is the sound still okay?
Yes.
Okay, great, thanks. All right, so we try to study simultaneously typical adults, typically developing children, and clinical populations using a multimodal neuroimaging approach, which includes task activation, fMRI, resting state fMRI, connectivity modeling, and diffusion tensor imaging to look at the structural connectivity underlying large-scale brain networks. And so I'll kind of go through some of the studies that we've done in these different domains and talk a little bit about how we think that working in these areas at the same time can help us inform development and aging and lifespan studies as well. And a lot of the work we do takes as a foundation network neuroscience, which is sort of a hot topic these days, but it's actually been around for even longer than neuroimaging has been around. So if you look at some earlier work from neurologists like Marcel Messelem, he's talked for many years about how cognition is subserved by interconnected neural networks. So any particular behavior is mapped at the level of multifocal neural systems rather than specific anatomical sites. And these are, insights that could have been gleaned from studies from neuropsychology, even before imaging became a tool of a popular tool to use in cognitive neuroscience. And there's work from the computational modeling, for example, from Randy McIntosh and others, that have shown how the functional relevance of a given brain area depends on the status of other connected brain areas with that area. So for example, here you see some networks in gray, and the idea here is that the function of that network might be sort of affected by what's going on in the interconnected nodes. For example, in panel A here, the black nodes might be co-activated, whereas in panel B, the black nodes down here are co-activated. So just the sort of context in which a network activates is also of great importance, and it's something that we try to keep in mind in all of our work. And finally, there's work by Luis Pessoa and others that suggest that networks need to be understood of the interactions between multiple brain regions as they unfold across time. So the time domain is another thing that we tend to, as much as we don't always focus on in functional MRI studies, it's a topic of great interest in our lab nowadays, is understanding network dynamics and how they underlie cognition. So these are sort of some of the background principles that we'll kind of keep in mind as we talk through some of the work I'll share with you today. Okay, let's see if I can get this guy to play. Oh, that looks like it's playing. And as many of you know, for years now, we've realized that the brain is never sort of just idling, and when you're not doing anything at all, there's these coherent fluctuations between brain regions that we call functional connectivity. So in this particular video that I steal from Daniel Margulies, you see that areas sort of come together at a very slow 0.1 to 0.01 hertz kind of oscillation. And the regions that you see, even though this is spontaneous intrinsic brain activity, it's not completely random, and it's not completely without some kind of organization. In fact, you can highlight regions that are involved in attention and memory and language and all kinds of networks that are involved in cognitive tasks. You can see correlations between these regions even in the resting state. So of course, we like to sort of exploit this property to understand the integrity of these large-scale brain networks and how they change across the lifespan. And one thing in particular that we've focused a lot on is trying to do studies that can span different age groups, so children, adolescents, and adults. But oftentimes when you're trying to study a high-level cognitive process like executive function or flexibility, cognitive flexibility, it's hard to find tasks that are applicable across different age ranges, as of course 7-year-olds aren't quite as capable of doing as complex things as adults. So there's been sort of a lot of ways to operationalize flexibility in the literature, some with sort of higher working memory loads and others that are a bit easier for children to accomplish. And we've sort of, over the years, kind of thought about this and flexibility in particular as a process that involves different executive functions, including attention, working memory, inhibition, and switching. And the nice thing about all of these processes is that years of task-related fMRI literature have suggested that there's specific nodes in the brain that seem to underlie these cognitive functions. And there's a diagram here from our 2015 review by Dina Dajani in the lab that just looks at the sort of frontoparietal networks that we know to be involved broadly in executive function, some with sort of more specific patterns of activation that highlight their unique role in supporting flexible behaviors, in particular the inferior frontal junction, or IFJ. And I'll talk about these nodes in the context of other sort of large-scale networks that we think are involved in more sort of general processes related to executive function. But this is just a summary diagram to show you some of the regions that we'll be getting into throughout the talk today. So one thing we tried to do is come up with a task we can use in the scanner that young kids can do, that adults can do, that can be sort of titrated in terms of difficulty. And so we took one that's adapted from Jackson Zalazo, the flexible item selection task. And here, it's an inductive task where you have to decide which category to group the objects by. So for example, we just asked the participants here, pick 2 items that go together. So you might say, well, you know, these 2 items, this set of boats and this rabbit, they're both blue. So I'm going to say that's one group of things that go together. And then we asked, pick 2 other things that go together. And so you actually have to, you know, the first part of the task is just an abstraction. The second is sort of inhibition and flexibility, because now we have to not think about this pair, but think about another pair. So maybe these 2 cards go together because they're both rabbits. And then you can think of what's another dimension by which these cards can be grouped. And you could say, well, in these 2 panels, there's 1 object. So that's another grouping. So you can make this task more or less difficult, as you can imagine, by adding cards and adding selections. And when this has sort of never been done in the scanner before, so to adapt it to the scanner, what we did was just create a control condition. So here, we just tell the participants, press the 2 buttons that are highlighted, and then press the next 2 buttons that are highlighted, and press the next 2 buttons that are highlighted. So this is a way of sort of controlling for the visual and motor responses that would be required for this task. So in just a group of 30 or so adult subjects, 18 to 40 something, you can, this is what the task looks like when you actually do it in the scanner. We give the people about 8 seconds to make 3 selections. And in the sort of control condition, they're just following along, pressing buttons without having to think about what they're actually doing. So if you look at the behavior, obviously we wanted to do some validations to see if what's been done outside of the scanner compares to what happens when you're actually laying down, pressing buttons inside the scanner. So if you look at a computerized version of this task and the scanner version, there's a very high correlation between the accuracy on the 2 versions. So that was good. People tend to get better or more efficient at the task as you go from run 1, 2, 3 to 4. So there's, if you use a combined reaction time accuracy metric called efficiency, you can see improvement on the task over time. And what's really interesting about this task is there's a lot of individual variability in the behavior, and it turns out a lot of individual variability in the neural responses during task performance. So if you just look at the task condition, the flexibility minus control trials, and this is again Dina Dajani's work from her dissertation, you see maybe what you would expect, a lot of frontoparietal activation, inferior frontal junction, inferior parietal lobule, cerebellum, basal ganglia, insula, and anterior cingulate cortex, a lot of widespread activation amongst the lateral frontoparietal and midcingulo-insular brain regions. And these are actually, the Z is like through the roof here. So they're very high levels of activation. And what's interesting here is that also, in addition to just the levels of activity, we're interested in the brain connectivity that sort of underlies the processing of this, this cognitive flexibility task. And as I mentioned, there's a lot of individual variability here. This is a directed functional connectivity modeling approach called group iterative multiple model estimation. And we collaborate with Katie Gates at UNC to use this tool. And basically just uses an iterative search algorithm that identifies the most parsimonious structural equation model amongst a user-defined set of regions of interest. So if you take all those sort of peak activations from the tasks that I showed you in the previous slide, and sort of look at how these sort of brain regions interact with each other during task performance, it turns out that the only region that's directly activated by the flexibility trials is the inferior frontal junction here in red. And all the other activations you see are sort of a downstream result of the IFJ influencing these other nodes of the network. And of course, there's a whole other story about the individual variability in these connectomes, which is interesting and part of the paper if you want to take a look at that. So we're using task-based fMRI to get at the sort of the regions that we think are critical for flexibility in adults, of course, first, and then sort of adapting this to the developmental paradigm. And we had to, of course, slow it down and give children more time in order to be able to complete this task in the scanner. But and sort of another line of work in the lab, we just look at development of flexibility by examining brain networks that we think are underlying these processes. As we know, there's sort of protracted developmental trajectories of cognitive flexibility and the executive functions that contribute to it. sort of evolve and expand over the adolescent years. We know from our previous work looking at effective and structural connectivity of brain regions underlying flexible behaviors that there's a change from childhood to adulthood where you see greater sort of influence of, for example, the insula to ACC and DLPFC when you compare children to adults. Also greater structural connectivity or fiber density between brain regions that are involved in some of these processes going from childhood to adulthood. If you just look at the Insula and ACC sort of Salience or Midcingulo-insular networks, they change in their spatial topography from the lifespan from age 10 to 80. They actually become more stable and more less spread out, if you will. And if you look at sort of interactions between networks, they change across the lifespan in ways we've looked at in several papers. And there's also a lot of interesting things about the signal variability in these brain regions and how they change from age 8 to 80 as well. So lots of sort of work in the lab just does this kind of general network characterization to see how the systems that we think underlie flexible behaviors change in their properties across the lifespan. So keeping that sort of in mind, here we're going back to our flexible item selection task and asking the question, how are sort of 9 to 12-year-old kids doing this task? And how are children with autism able to do this task? And what are the brain systems underlying that? So this is just a group of, this is our initial analysis of 9 to 12-year-olds doing the flexible item selection task. And you can see pretty good activations in frontoparietal, insula and ACC regions, just as we saw in adults. So this is a promising task, we think, that can be used across different age ranges. And this was something that we started a few years ago as a registered report. So we said, okay, we're going to collect data from 40 children with autism and 40 typically developing children. We're going to have them do the flexible item selection task, and then we're going to write the paper or submit the results. So as it turns out, our scanner went down on March 20th of this year and was replaced by a newer model. And at the same time, that last week of March was the last week we could have collected participants because of the virus. So we, obviously, as many people are now realizing, research has to take a backseat when something like this comes up. So we have fewer participants than we would have liked in this final data set. But these are sort of the dangers of registered reports. So we'll see what happens with this. But in any case, we are hoping to compare the children with autism to the typically developing kids to see if they actually are using these brain systems differently to accomplish the task. So let's see. Oh, okay. So here, basically, what I've shown you is that we think that cognitive flexibility involves coordination among brain regions supporting inhibition, working memory, attention, and salience detection. Adults and children both seem to engage lateral frontoparietal and salience, or what I like to now call midcingulo-insular networks, during flexible item selection. And I'm figuring out how to scroll here. And task-modulated connectivity of the inferior frontal junction seems to be related to flexible behaviors, at least in this task setting. So stay tuned for that pre-registered data from children with autism spectrum disorder, which will help us understand the question of how these systems are disrupted in a neurodevelopmental condition where we see inflexibility. So another thing that we think underlies flexible behavior is brain dynamics, as I sort of alluded to in the beginning. And there's several ways to think about brain dynamics, and there's one that's sort of become quite popular in the neuroimaging literature, and that's a sliding window functional connectivity analysis. So here, instead of sort of collecting data for 10 minutes and then averaging the connectivity between different brain regions over that 10-minute course, what you can actually do is use windows of 45 or 30 seconds in length and look at how functional connectivity across the whole brain changes across however long you have of a scan. So you can use then something like k-means clustering to figure out if there's repeating patterns of functional connectivity that reoccur across the entire duration of the scan. So you might have a particular brain state or functional connectivity state that you label, and then another one, and then another one, for example. And what you can then do is sort of quantify how the brain toggles between these different configurations. Of course, these correlation matrices are just an example here, but it's if you took every brain region by every brain region as determined by some parcellation scheme, you can see that there's changes between these whole brain functional connectivity maps that occur across time. And you can quantify, for example, how frequently does one brain state appear. You can quantify the dwell time, which is how long does a brain stay in one state once it gets into it. State transitions are an index of how many switches there are between distinct brain states. So this is something we've been playing around with, and others, of course, have been doing for the last few years. It turns out that, and this is work by Jason Nomi in the lab, if you look at the nice big data set of the Human Connectome Project, this was a few years back when there was a couple hundred subjects available, but these subjects had an hour of resting state fMRI data, which is really great for questions of dynamics. And the subjects are also have undergone all these different neuropsych tests to determine things like attention, flexibility, working memory, and fluid intelligence. So lots of data here that I'm sure many of the audience is familiar with. So if you actually take these data sets and use something like independent component analysis to get sort of whole brain parcellation. Oh, I see a chat question. Should I be looking at that? Oh, so should I answer questions as we go, or should I wait till the end?
Our plan was originally to wait to the end.
It says urgent, so it might be on, for those who flag for urgent, I think that means that they want it addressed or we can't really move forward.
I see it now. So it says, how would the increase of salient causal outflow with age play out in the flexibility paradigm between young and old cohorts?
Yeah, that's a good question. We haven't looked at this yet in our younger data sets. So the flexibility outflow change, or sorry, the causal outflow changing with age is something I showed you in a resting state fMRI, and we have yet to conduct that analysis in the task data set. So that's definitely on the horizon. Great. So basically, yeah, so we're looking here at how brain dynamics sort of may relate to individual differences in flexible behaviors as measured outside the scanner. And so in this work, what Jason did was just take the subjects and take their huge resting state fMRI data set and sort of decompose it into different functional connectivity states. And here I've just ordered them by those that occur the most frequently and those that occur the least frequently. And one thing you might already notice is that states that occur very frequently are characterized by sort of a whole brain sort of weakened functional connectivity pattern where there's not sort of strong dominant patterns of connectivity between brain regions, whereas the states that are less frequent show pretty strong correlations between brain regions, but they don't occur that often. And it turns out that those individuals who show more sort of occurrences of state 1 and fewer of state 5 are doing better on working memory. And a similar finding was shown for the cognitive flexibility, whereas where those individuals who spend more time in state 2 and less in state 3 tend to perform better on the Wisconsin card sort variant that's in the HCP data set. So there's some suggestion here that greater cognitive flexibility, in adults at least, is associated with a propensity to occupy more frequently occurring brain configurations that are characterized by attenuated correlations and greater functional connectivity variability. And this is just showing you one way where you can take sort of whole brain dynamic approaches to understand how brain dynamics might be supporting individual differences in flexible cognition and behavior. So that's sort of the whole brain story, but we, for a long time, have been convinced that there's a few sort of networks in the brain that seem to dominate across all kinds of cognitive paradigms. So if you're, do sort of meta-analyses, or you just look across different types of tasks, you might have come across these terms and these networks before. So of course, there's the default mode network with nodes in the ventromedial prefrontal cortex, posterior cingulate, which I'm trying to now get people to call the medial frontoparietal network, just so we get away from task terminology and more towards anatomical terminology. And then the Salience network, which I'm now trying to get people to call the Midcingulo-insular network, with key nodes in the Insula and ACC, and the central executive network, which I'm hoping people will call the lateral frontoparietal network, which we know to be involved in executive function, working memory more broadly. And what's been interesting over the years is noticing that there's relationships between these networks. So when central executive goes up, default tends to go down. And if you do, causal analyses using Granger, DCM, or what have you, you often find that you can predict what's going to happen in the other two networks from what's happening in the Salience network, in particular with the dorsal anterior insula seeming to have a special role here. So kind of thinking about these large-scale brain networks, we're interested in whether dynamics of some of these systems relates to individual differences in behavior as well. So this was a project in collaboration with a group in Berlin who conducted a task, which should have been a very easy task, but there's some distractors, and I'll show you what that distractor looks like. So if you look at the screen, the task is just to say if the letter E or F pops up, and so there's an arrow that sort of cues you, and you're supposed to look over to that side of the screen. And then if we use eye tracking, we can see whether people actually did that or not. At the same time, there's going to be a distractor, and it's sometimes much more distracting even than what I've shown you here. But basically, the idea is that people often move their eyes over to that distracting image and miss the target, which they were supposed to be monitoring. And you can use eye tracking to see exactly how badly they missed the target. So you can actually quantify that. And it turns out that, as you might expect, when there's a sort of exciting erotic distractor, people are very bad at the task of detecting the letters. Their error rates go up, and they look away from the target. You can do all kinds of quantification of their eye gaze behavior, which is really interesting because people might report, or they might say they weren't distracted, but their eyes sort of tell a different story. And it turns out if you look at the dynamics of this network, which we've called the Salience here, Insula and ACC-based network, there's one sort of configuration of this network, a dynamic configuration that does relate to behavior on this task. So individuals had resting state fMRI collected separately and then completed the task I just showed you on a different day. There's about 100 college students in Germany. And Rosa Stemke was the first author of this paper. And it turns out that the individuals who spend more time in this state, which is state 4 of the Salience network, when these regions, Insula and ACC, are negatively correlated with the visual cortex, this particular frequency of occurrence of this state was actually related to the eye gaze behavior. So the ones who spend more time in this state were actually able to do the task better. And we speculate that that's because their visual cortices weren't as coupled or as communicative with the Salience network here. So it suggests to us that specific network dynamics like the Salience, Midcingulo-insular in particular correlate with the capacity to resist tempting distractors and self-control behaviors generally. And this is a sort of fun way of demonstrating how these sort of intrinsic network dynamics might be related to individual differences in self-control. So if you really are interested in the insula, which we are, you'll find that it's one of those regions which shows up in so many ways across so many tasks that it's hard to understand what this brain region is actually doing. But if you look at meta-analytic studies, emotion, empathy, and pain, sort of affective processes, tend to strongly activate the insular cortices. Awareness, interoception, somatosensory, and sort of bodily states, all of these things activate insula. But high-level processes like conflict, switching, attention, and inhibition, those things we talk about when we talk about executive function, also very much activate the insular cortex. So it's one of those does everything brain regions. And the truth is, if you look carefully, there's a reason why it seems that the insular cortex does everything. And that's because, of course, it's probably not one homogeneous cortical area. There's anywhere between 2 and 27 subdivisions within the insula, depending on who you ask and what parcellation you look at. Here I'm just showing 2 studies which suggest a tripartite subdivision with a sort of dorsal anterior insula, ventral anterior insula and a posterior insula. And these types of studies, you just, in resting state fMRI, you ask the question of the voxels of the insula, what other brain regions are they connected with, functionally connected with, and then you sort of cluster the regions based on similarity of functional connectivity with the whole brain. So I'm just showing a couple examples from Ben Dean and Luke Chang where they suggest that at least 3 subdivisions can be found. And if you look at meta-analytic connectivity modeling and types of analyses where you look across hundreds of task activation studies, there seems to be a few patterns that emerge. So studies that report activation of the ventral anterior insula tend to be reporting paradigms involving emotion and affect, like as you can see on the left from Luke Chang's paper. These are terms that come up in those papers where ventral anterior insula is activated. And on the right is some work we did with Luis Pessoa and Michael Anderson a few years ago, showing, this is the meta-analytic connectivity modeling, showing that when you have ventral anterior insula activation, you tend to also get co-activation in orbitofrontal and some limbic regions. Whereas when you get dorsal anterior insula activation, that shows up in studies where the terms on the left in blue, cognitive error processing, inhibition, switching, are in the papers. And the co-activation network of the dorsal anterior insula tends to involve frontoparietal association cortices. Whereas the posterior insula looks to be co-activated with somatosensory regions, and also in studies where you are looking at pain, stimulation, all of the terms over here in green on the left. So there's some evidence, at least, that there may be some partitioning within the insular cortex if you look at it this way. And at the same time, there's a common functional fingerprint where we found that basically across all of these task domains, all of these ROIs within the insula are activated. So there's sort of some specificity, but also some generality to the subdivisional structure here. It turns out, so again, Jason Omi in the lab was looking at dynamics of the insula to try to answer the question, why are they so involved in everything, these subdivisions, and why are they sometimes differentially involved in tasks and sometimes commonly evolved across tasks. And on the left here is just a kind of polar plot depicting each of the insula subdivisions you can see up here derived from an independent component analysis. Oops, okay. And the long story short is that you can get different states of functional connectivity using the dynamic approach I mentioned before. And in some of these states, the insular subdivisions show similar patterns of functional connectivity with the rest of the brain, like for example in state 3. Whereas in state 2, the different insular subdivisions show divergent patterns of functional connectivity. So the question of what is the insula talking to, it depends on what time window you look at. So some regions are sort of talking to the whole brain more often than other subregions. And what's interesting, if you kind of quantify the state, sort of the different states that each insula subdivision occupies, the dorsal anterior subdivision is the one that shows the most variable functional connectivity pattern between states. So state 1 is different from state 2, is different from state 3, et cetera, et cetera. So even though all the insular subdivisions seem to show variable patterns of whole brain functional connectivity, the dorsal anterior insula seems to be the most variable in its connectivity partners. And then if you look again at that dorsal anterior insula subdivision, again, this work from HCP by Jason Omi, you'll find some unique structural connections associated with that insular subdivision. So especially with the frontal cortices, lateral frontal cortices, and thalamus, there seems to be some structural connectivity there that we don't see for the other insula subdivisions. So this is all our sort of, I guess, segue into the insula because we love it so much, but it's just to show you that there's a lot of interesting things once you sort of look into that cortex and figure out what's going on there. In adults, at least, you can subdivide the region into a dorsal anterior, ventral anterior, and posterior subdivision, with the dorsal sort of leaning towards more involvement in cognitive control processes, the ventral more in the emotional and affective domain, and the posterior a little bit more in the somatosensory function. And what's interesting about all of this is the dorsal anterior insula seems to be unique in its variable functional connection patterns and its structural connectivity that we think might permit functional flexibility that we see of this region in the task setting. And after years of doing this, we actually went back, I went back and looked at the effective connectivity of the dorsal versus ventral insula. I don't think I ever published this, or maybe I don't remember. But basically, the dorsal anterior insula is the one that shows the sort of causal effect on the other networks. The ventral anterior insula doesn't show the same effect. So there's something really interesting about what the dorsal anterior insula is capable of doing in terms of orchestrating network dynamics. So at the end of the day, it's hard to tell what's going on where with fMRI. So a lot of work has started to focus on using intracranial recordings in patients who are being monitored for epilepsy. So I'm showing some work from Rick Betzel's group and Aaron Cusey. They're sort of applying sort of network metrics to data that's derived from the cortical surface to understand relationships between brain networks at a very fine temporal resolution. Of course, you can get millisecond resolution here. And so what we're excited about is a collaboration now that's sponsored by the Canadian Institute for Advanced Research, where we're looking with surgeons at the University of Western Ontario who routinely implant depth electrodes in the insula. And I was surprised to find that this group often does up to 3 depth implantations with anterior, mid, and posterior insula locations. And what we're doing now is developing a battery of tests that we think will help us to really understand the sort of timing and the function of these different insula subregions with a much greater amount of much greater temporal resolution than we used to be able to see. So look for that in the future. And finally, all of this sort of work in adults and typical development helps us to better characterize, we think, better characterize clinical populations where we see alterations in flexible behaviors like autism. And one thing in particular that always surprises me is these sort of striking statistics about outcomes, adult outcomes in autism. So about 80% of children who grew up with autism end up living with parents or guardians, sort of not leaving the home, not living independently, which is a very high percentage compared to other developmental conditions. And also 80% of individuals with autism, young adults with autism, tend to be underemployed or unemployed, which is also very low compared to what you see in learning disability and intellectual disability. And I think we always tend to focus on the social communication deficits in the disorder, which undoubtedly contribute to some of these grim statistics. But another thing we often sort of don't realize is important is the flexible behaviors are really what's necessary for transition to adulthood in many ways. And some targeted treatments for executive function sort of bolstering and executive function training do look at improving flexibility specifically. So I think that's sort of a future direction in terms of thinking about how we can, help individuals with autism, maintain employment and independent living, as they transition into adulthood. One thing we've noted, though, is that not everyone, of course, with autism shows the same pattern of executive function deficits. And if you use something like latent profile analysis, like this is again some work Dina Dajani did in collaboration with Stuart Mostovsky's group, and he's got a great data set that includes autism, ADHD, and comorbid autism and ADHD. And long story short is that not every child with these disorders falls into an impaired executive function group. In fact, the group of individuals with impaired executive functions includes individuals with ADHD, those with autism, and a really large proportion of kids who have comorbid disorders. So that seems to be sort of a double hit in terms of executive function deficits. But there are, if you look in the average executive function group, a good amount of children who children with autism, about 18% have average executive function. So it's not that everyone is equally impaired. And we replicated this. Adriana in our lab was an undergrad honors thesis, published a replication of this original finding, where again, like here, we saw about a third of our kids with autism fell within the average executive function group. So some kids with autism will require, additional training, additional sort of, cognitive or any kind of bolstering in this area, but not all kids will. So the sort of stratifying this heterogeneity is one of our goals, and we do this both on the behavioral and the brain level. At the time, I'll talk about that later, but looks like we're running short. But of course, we want to understand the neural systems that underlie these individual differences in flexible behavior. And the first hypothesis we had was that maybe in autism, the brain state transitions are actually reduced such that there's just fewer sort of transitions between brain states in autism, and this might underlie cognitive inflexibility. There's been a few papers published by a lot of people scooped us on this. So there's a handful of papers that have shown this by now. In our own preliminary data, looking at some samples from the Autism Brain Imaging Data Exchange, we did find overall a reduced number of transitions in the individuals with autism compared to the typical individuals. We also are now starting looking at different kinds of analysis for brain dynamics, which is called co-activation pattern analysis. Here, there's no need to specify a sliding window arbitrarily. Instead, we just take a region. For example, here, we were interested in the Salience or Midcingulo-insular network. And if you look at the time course of that set of regions and pick the ones where they're sort of most active, then you sort of cluster the timeframes that are most active, or you take the timeframes that are most active across the whole brain and cluster them by their spatial patterns, and you can get what are referred to as co-activation patterns that show you what other regions are sort of highly active when these regions are active. And it turns out in this study by Emily Marshall, another honors thesis in our lab, she actually found these sort of co-activation patterns, labeled them 1 through 5 here. And the group difference, and of course we focused a lot on the Insula for all these reasons I mentioned before, but what we found is that across these states, there weren't a lot of differences in the expression of these co-activation patterns, except for in state 2, where typical individuals, typical kids entered that state more frequently than the kids with autism. And state 2 is a configuration in which the Salience network is actually co-active with parts of the frontal parietal systems and parts of the default mode network. So it's kind of a co-activation pattern amongst all networks that seems to be more available in the kids who are typically developing than in the kids with autism. So these are some initial ways of sort of teasing apart how brain dynamics relate to disorders and how they relate to flexible behaviors. So stay tuned, I guess, on this one too. So dynamic functional connectivity approaches reveal atypical patterns of brain dynamics, particularly reductions in transitions between states in neurodevelopmental disorders that we know are characterized by cognitive inflexibility. And the extent to which these differences these dynamics underlie individual differences in flexible behaviors is what we are currently working on. And this is sort of the ultimate goal that many of you have heard the term precision medicine. There's a review I have with Manish Sagar at Stanford where we talk about what we think would need to happen for neuroimaging to get towards contributing to precision medicine in psychiatry. So there's heterogeneity, there's a lot of issues I've brought up so far, but we think that psychiatric neuroimaging needs to move towards ecological validity, reliability with data sharing is a big part of that. Understanding mechanistic, sort of models of computational modeling of neural processes and, translation to the clinic. And we think we're actually a far way off from this. So this is kind of more speculative future thinking for where the field might go. But if anyone who's been paying attention has heard of the RDoC sort of initiative from NIH, and the idea is sort of rethinking DSM-based categories that are sort of symptom-based that might include very heterogeneous groups of people, to stratifying individuals along a continuum and also based on biological and other factors, other sort of biological markers, to get data-driven categories that might be more homogeneous and thus more amenable to particular treatments. So the reason I show actually the Insula and Cutbirth paper from 2015 is mainly because I think it's cool that they just happened to mention the Insula in their figure. And I don't know if they meant that because they think the Insula is important or if they just did that for fun. But I think it would be really based on what we've been looking at for the last 10 years. The insular cortex does seem to be important across a range of neurological and psychiatric conditions, neurodevelopmental, neurodegenerative. It's just one of those areas that seems to be involved in so many different functions and dysfunctions that I think they're right to put that in the brain image, in their figure, even if they didn't mean to. So all of this is to say that we are, interested in understanding flexible behaviors in the brain systems and dynamics underlying them, with the ultimate goal of sort of, finding an application in autism to sort of get kids to, worry less about what's on their feet, for example, and be more, focused on their interactions with the world. And yeah, in the interest of time, I think what I'll do is I'll stop there and go into the question mode. And I can show additional sort of follow-up data if anyone's interested. I always have backup slides, so I'm happy to stop there and take questions. Thank you.
Thank you. I actually have a question coming in.
Okay.
Okay. Regarding dynamic network analysis, temporal, do you think fMRI data provides sufficient temporal resolution to probe the neural activity in real time? Can you comment on this? Thanks a lot for the excellent presentation.
Yeah, that's a great question. Yeah, of course, fMRI is slower than we would like. For years we've been working with TR of 2, so sampling rate of every 2 seconds. What's nice is that we are seeing data sets released now that are a bit faster, and then we're seeing multiband, we're seeing, TRs are dropping, so you're getting slightly better temporal resolution than we used to have. In terms of the sort of causal modeling and all of these kinds of issues, it's always a question of, well, is that enough to account for the HRF, hemodynamic response delay differences between different parts of the brain? And that answer to that depends on who you ask. So I think for a lot of this work, it's nice to see multimodal convergence. It's nice to see different kinds of causal models converge. And so fMRI obviously isn't the best tool for this, but it's sort of the tool we have at the moment.
Thank you. And actually, at this time, before we continue, I'm going to unmute everyone so that we can give a nice round of applause for a wonderful presentation. Thank you so much.
Thank you.
Great talk.
Do you want to resume with questions, Lizzie, or do you want? I'll take over from here.
Sure.
Is that the one? So you did the one from Yin Wang?
That's correct. I'm going to mute everybody again except for our speaker and Fumiko.
All right. I think people are still typing. I think there's just one. As we are waiting, does anyone want to chime in while we're waiting for someone to type? Does someone want to chime in verbally?
I have a quick question if there's no one else.
Well,
Sure, go ahead.
Thanks, Lucina, for a great talk. I was kind of just thinking about the inferior frontal junction and its sort of origin for directing the other regions in the initial causal connectivity study that you showed. And then you, I think, mentioned that the dorsal anterior insula might also play that role. And I was wondering whether that is related to the brain state that it's in and whether you've looked at that, and whether it's also related to switching between other brain states. So for example, I think the one that you said was the dorsal anterior insula in the midcingular network connecting with the frontal parietal, and whether there's subsequent networks that, or subsequent states that those networks happen to also be connected in. For example, maybe the frontal parietal network and the visual cortex in the visual network.
Yeah, this is a great question. And it, for a long time, I was sort of focusing on resting state networks and resting state sort of configurations. And then as we sort of got into the task domain a little bit more, tried to understand, how do, these systems respond in the, in these different kinds of conditions, we realized not everything you see in the rest state directly translates to what you see in the task state. So even if the, the insula driving occurs, in multiple studies we've seen in the resting state, there could be other drivers in task conditions like the inferior frontal junction, for example. So the truth is our lab doesn't do a lot of tasks. So I think this is great sort of follow-up work for anyone who has a bunch of task data and wants to systematically explore how these different network interactions sort of change across different tasks. The HCP data set is one of the great ones that we've sort of started, and everyone started to work with, on these topics. But I kind of hope that other people will jump on and just kind of do a bunch of these analyses. Nowadays, the data are all out there. The code is even all out there. So it's just a matter of time.
Ted Rosenkrantz from UConn. So the clinical side of me is a neonatologist. And we've known for quite a while that our preterm population, particularly the boys, have a fairly high rate of autism. The other thing we know from some of the work done down at Yale, I think Laura Ment's involved with that, is when you start looking at connectivity of the preterm babies compared to the baby at term and adjusted for the prematurity. I mean, circuitry is all different in the preterm baby. And now I'm starting to think, well, this is certainly, if the circuitry is changing around, this is certainly plenty of opportunity to have other miswiring or wiring that's not correct that may contribute to what we're describing as autism, but could also be different circuitry than the baby born at term who turns out to have autism. So it was just a fantastic talk that really got my mind going now. Thank you.
Well, thanks so much for that. I actually have been thinking a little bit about this because someone asked us to write a review on risk for autism. And of course, there's these infant sibling studies that are now being done to sort of, the kids who have an older sibling with autism to see if they go on to develop. And so there's quite a emerging neuroimaging literature looking at the sort of brain networks that could possibly, or the sort of configurations that could possibly predict going on to develop autism. And I'm happy to send you that if you're interested.
Yeah, I was going to say the other piece, so you're down in Miami? Because Eduardo Bancalari is the head of the neonatal unit there. Fantastic guy. But that might be a source of preterm infants who you could start comparing to term infants, even as they get older and whatnot, and follow what's happening with the autistic, non-autistic. I mean, if you're looking for candidates to enroll in your studies. So.
Yeah, thanks very much. I'll keep that in mind as if we ever get out of our homes to try to drop a visit over there.
Sure.
Thanks very much.
Hi.
I think I see some questions on the side. Should I go to those?
Yes, I'm gonna read out the slides, and then in the meantime, I'll put one slide up explaining about the BIRC Speaker Series, just for those who joined later, and then how to join the list server for the future. So this quick question is, I was just wondering about the purpose of the deep electrodes in the Insula from the Canadian group.
Yeah, that's actually related to the previous question about temporal resolution with fMRI. So because we know that we're getting this sort of lag in the blood oxygen level, the bold response when we do fMRI studies, the question is always, can we sort of trust the timing information from that data? And the only way we can sort of trust the timing of events is with the deep electrodes in the insula, which are measuring sort of directly from the cortex. So it would be able, once we have a good data set collected, we'd be able to see when exactly does the anterior insula come online, and how does it precede activity in other frontoparietal regions, for example. So that's more of helping us answer the timing question. And should I go on to the next question on the chat?
Sorry, I keep on forgetting to unmute myself. Yes, I will read the question.
Okay.
Then I don't see a lot of people doing dynamic connectivity analysis or other connectivity methods. More often applied in resting state data on task fMRI data. Do you think it's okay to do this, or do you know why this is? For example, doing sliding window on a block fMRI design.
Oh, yeah, actually, I don't see that question in here. Is that a, okay, well, okay, so, just using dynamic analysis on task design, is that the general question?
Yes.
Okay, yeah, I think, yeah, people are definitely starting to do it. It's more of a it becomes like a little bit methodological in the weeds question of when you're looking at a resting state paradigm where there's no task versus a task paradigm where there's onsets and offsets and things. There's a lot of work from Michael Cole and others showing that the connectivity estimates are sort of inflated by the onset of the task and things like that. So there's special sort of deconfounding strategies you can use when you're trying to compare rest and task data sets. Then there's other work from like Bharat Biswal's group that suggests that, you know, when you're doing a task rest comparison, you may or may not want to regress out the task waveform. And so I think there's just more of at this point, a lot of methodological disagreement about how to think about task and rest connectivity and how to think about task and rest dynamics. We have a paper out, what's his name? Reed is the first author and Michael Cole is the last author. It's in Nature Neuroscience that kind of gets out all of these questions about connectivity and how to think about it conceptually. So I can hopefully share that link if anyone's interested in a really deep dive into that great question.
Maybe we'll get that from you and then we'll distribute it to the registered people. Thank you.
Sure.
The next one is, since autism is known to have comorbid anxiety, what kind of temporal dynamics between insula and amygdala have been observed?
Yeah, that's a great question. We haven't looked at the comorbidity with anxiety in our own studies. For sure it's there in probably like 30% of kids with autism, I understand. So I don't know if any studies have looked yet at the insula amygdala sort of dynamics in autism, but the truth is anyone can do this. Just, you know, download the ABIDE data sets that I mentioned that Adriana DiMartino at NYU has gathered. And those are all resting state fMRI data sets, but I think that's a question that is empirical and could be answered.
Great, thank you. Since you mentioned the pitfall of using fMRI due to HRF lags, could you comment on how to take care of using GC and avoid misinterpretation of the direction of the flow?
Yeah, I mean, the truth is I haven't done GC in years because of this exact problem, is that you can't like entirely avoid the misinterpretation. The one thing that keeps me sort of a little bit faithful is that a lot of the results that we've seen pertaining to the insula are still there with DCM and other sort of state space models that don't look at just the, that aren't just sort of the time series models. So if you believe that using multiple methods and multiple approaches and getting the same result is a good sign, then that's sort of, but yeah, Granger has that problem. There's been many papers written about these problems and how you can't ever really, ever disentangle that if using just Granger on the time series data. So I just try to use other sort of, or look at complementary approaches and see if you converge on similar findings.
Great. Clinically, why do they insert deep electrodes in the insula?
Yeah, people are really interested in that. Yeah, it's actually just a thing that's only done in the case of severe and intractable epilepsy. So it's for monitoring where the seizures originate, because at some point surgeons will go in and remove some cortex if they're really causing large seizures. So they basically, and really as a worst case, last resort kind of scenario, they'll implant these electrodes to try to monitor those conditions.
Great. How does puberty affect task switching and connectivity in both the ventral and dorsal insula? Really awesome presentation.
Thanks. I wrote a grant to study puberty and development in autism 5 years ago. It was never funded. So I don't know, but I think that, and I think a lot of the people who are more seasoned developmental cognitive neuroscientists than me will tell you that pubertal hormones and development have a lot to do with brain organization and network organization. And in fact, maybe even more so than just chronological age. So I think that it's a very important question. I don't know if anyone has addressed the more general questions about how puberty affects brain network development, more broadly. So I think this is just an open question, active area of research. If you find out the answer, please let me know.
Great, thank you. So I think that's it for questions. I wanted to thank everyone, especially the speaker, Lucina, for joining us today. Again, this is a unique kind of fun way we're trying to do the Speaker Introduces Speaker series, where we have some names from Lucina, and so we will get the date and speaker on the schedule. So please join us on our listserv if you'd like to join us. It gives me a reason to put up makeup once in a while, every several weeks. So I think this is a great one for me as well.
I didn't even bother with that, but.
So thank you very much, and we look forward to seeing you around.
Thank you so much, and please do email me with any follow-up questions. I'm happy to be in touch with anyone who wants to be in touch. So thanks very much, Fumiko.
Thank you. Thanks, Lucina. Thanks,
Thank
everyone.
you all.